This presentation contains our discussion for Chapter 19, the family Enterobacteriaceae. Here is the outline of our discussion. As usual, we'll start off with the general characteristics applicable to every member of Enterobacteriaceae. Then we'll go on to discuss the opportunistic Enterobacteriaceae species. We'll be looking further into the different strains of Escherichia coli before moving forward to the other clinically significant opportunistic species, as we can see here in this second column. And then after that, we'll take a look into the true pathogens from the family. And lastly, alamin po natin kung alin-alin yung mga culture media commonly used in the isolation of Enterobacteriaceae. Let's now begin with their general characteristics. They are facultative anaerobes, which, in case you forgot, means that they can grow either in the presence or absence of oxygen. They are also non-spore-forming, gram-negative bacilli. So all members of Enterobacteriaceae are glucose fermenters. So if the organism was not able to ferment any of the sugars present in the TSI, then this is most likely not a member of Enterobacteriaceae. Next, all members of Enterobacteriaceae have pyrrhichircous flagella. All of them are motile at 35 degrees except for 3 members, which are Klebsiella, Shigella, and Yersinia. So Klebsiella is one of the opportunistic pathogens, while Shigella and Yersinia are true pathogens. So later, as we go through our discussion, i-explain ko rin po what we mean when we say true pathogens versus yung opportunistic enterobacteria lang. Next, all enterobacteria are non-encapsulated except for 2, and that is Klebsiella and Enterobacter. Klebsiella and Enterobacter have capsules. And then under microscopic examination, after gram staining, all members of Enterobacteriaceae can be seen as straight, gram-negative rods with rounded ends. So as I proceed with the discussion, hindi ko na po ipapakita ulit yung microscopic characteristics nila individually because they all look like this. Okay? So magpapakita na lang po tayo ng microscopic morphology nila if ever they have any special characteristics. There are 3 antigenic determinants that can be used for the serologic identification of infections that may be caused by enterobacteria, and these are antigens that can be found on different parts of a bacterial cell. So one of these is the somatic antigen, which can be found at the cell wall. So the somatic antigen is otherwise known as the O antigen. This one is heat stable, which sets it apart from the other 2 antigens kasi po itong dalawa, they are heat labile, meaning that they can be destroyed if the sample is subjected to heating. So in some serologic methods, if they want to increase the specificity of the test in detecting the O antigen, they would subject the sample to heating, which would eliminate the interference of the 2 other antigens. And there you have it. Yung somatic O antigen na lang po yung madedetect natin. Take note that the somatic O antigen is used in the serologic typing of Escherichia coli and Shigella species. Next is the flagellar H antigen. So as I've already mentioned, this one is heat labile. It can be found on the flagella of the bacterial cell, as the name implies. So the H antigen is used for the serotyping of Salmonella. And then the third one is the K antigen, or the capsular antigen. So this is also heat labile. Now for different species, may kanya-kanya din pong pangalan ang K antigen. So for Escherichia coli, this is referred to as the K1 antigen. Para naman po sa Salmonella enterica, the K antigen is called the VI or V antigen. So please take note of that. Here is an illustration pointing out where each of these antigens can be found. So specifically, saang parts po sila ng bacterial cell makikita. So if you're a visual learner, I hope this helps. Now, on to the opportunistic members of Enterobacteriaceae. Let's talk about their associated infections, as well as their colony morphology, and some biochemical test results. So when we say opportunistic members, all of these species are actually a part of the normal flora of humans. And as with all other opportunistic organisms, these do not generally cause disease in healthy, immunocompetent human hosts. Do take note, though, that they still have some significant virulence factors and may cause extraintestinal infections, which are observed outside of their normal body sites. Escherichia coli, commonly known as the colon bacillus, is the most clinically significant species in this genus. So may mga iba pa pong species na kasama sa genus na to, such as Escherichia hermannii, Escherichia vulneris, and Escherichia albertii. But again, ang focus muna po natin is dito lang sa Escherichia coli kasi dito pa lang ang dami na natin kailangang aralin. So in water testing, or the bacterial analysis of water purity, which is usually done for water supply companies and water refilling stations, malalaman po natin dito kung possibly contaminated ng fecal material yung water supply if we were able to isolate Escherichia coli from the sample. So yun po yung ibig sabihin nitong statement na to. Okay? So Escherichia coli is a primary indicator of fecal contamination in water purification. Now, clinically speaking naman po, Escherichia coli has been documented as the leading cause of nosocomial urinary tract infections. So Escherichia coli has both types of fimbriae or pili, and these serve as its virulence factor. So meron po sila both ng conjugation pili, and they also have the common pili or the adhesive fimbriae. So the common pili, this helps the bacteria to attach to surfaces. So other examples of bacteria that have the common pili are Neisseria gonorrhoeae and Pseudomonas species. The other type of pili is the conjugation pili or sex pili, which is used by other bacteria during the process of genetic transfer, specifically conjugation. Okay, so recall po muna natin yung process of conjugation. During conjugation, one bacterial cell can transfer or donate some of its genetic material to a recipient cell. Ang problema po dito, the donor cell contains a DNA, or if, for example, that donor cell contains a DNA that carries the genetic code for antimicrobial resistance, pwede niya po itong ipasa to other bacterial cells that do not yet have the code for antimicrobial resistance. So isa po ito sa mga ways on how multi-drug resistant organisms are spread. Okay, so as previously discussed in your laboratory classes, specifically on chapter 9, alam nyo na po yung meaning ng INVIC, di ba po? So indole production positive, methyl red positive, Voges-Proskauer negative, and citrate negative po ang INVIC reaction ng Escherichia coli. And then ito naman po yung TSI results niya. So acid over acid, and also positive din siya for gas production, but negative for H2S or hydrogen sulfide production. Okay, so after we take a look at the colony morphologies of Escherichia coli on different culture media, isa-isayin din po natin tong mga strains na to. Okay, so on blood agar plates, most strains of Escherichia coli are non-hemolytic, and they look like this. Okay? So some strains have been described as beta hemolytic. Meron din daw pong mangilan-ngilan na strains ng E. coli that showed to be beta hemolytic. But in general, okay? In general po, non-hemolytic ang karamihan ng Escherichia coli. On McConkey agar, they are seen as flat, dry, pink-colored colonies, also with a pink discoloration on the agar in their immediate surrounding, which tells us that they are lactose fermenters. So although there are a few strains that have also shown to be non-lactose fermenters. Again, majority po ng Escherichia coli strains are LF or lactose fermenters. So as we can see here in this picture, ayan po, these isolates on the top are an Escherichia coli that is shown to be lactose fermenting. So, maliban sa pink, yung mismong color ng colonies nagkaroon din po ng pink precipitates or pink discoloration sa paligid nila. Pero kapag naman po non-lactose fermenting strain yung na-isolate nyo, ganito po yung magiging itsura nila. So, usually they're colorless and they also will not produce a discoloration or a precipitate around their growths. Next, on eosin methylene blue agar, which we'll talk more about later, dito po natin makikita yung characteristic greenish metallic sheen ng Escherichia coli. Recall time? Recall muna po tayo, which non-fermenting gram-negative bacilli has also been described to produce a metallic sheen on blood agar plate. Okay, so ano pa po yung isa pang bacteria na nagpo-produce din ng metallic sheen, pero sa BAP naman? The correct answer is Pseudomonas aeruginosa, as we can see here on this pop-up picture to the right. So, ang ganda, no? Ang ganda nila. So, wala po. Natutuwa lang ako pag nakakita ko ng mga ganyan, mga magagandang colonies. Just, microbiology is so beautiful. So, again, on BAP po, ang may metallic sheen colonies ay si Pseudomonas aeruginosa. Pero kapag sinabi naman ng EMP, ang may greenish metallic sheen po dito is Escherichia coli. Next, on XLD or the xylose lysine desoxycholate agar, na madidiscuss din po natin mamaya, Escherichia coli will produce large, flat, yellow colonies as we can see here in this fourth picture. Let's now discuss the different pathogenic strains of Escherichia coli. First up is the uropathogenic E. coli or UPEC, and this is the most common cause of urinary tract infections in humans. UPEC possesses a type 1 fimbriae which adheres strongly to the urinary tract of the host, making it very difficult to wash out simply by urination. Next is the enterotoxigenic E. coli, or ETEC for short. So this has been associated with what is known as traveler's diarrhea. So nabanggit po natin kanina, di ba? E. coli is a primary indicator of fecal contamination of water sources. So now, the reason why it was referred to as traveler's diarrhea is because when people travel, say for example, to third world countries or to specific regions that are very poor and hindi pa po ganun ka-develop yung mga water purification systems nila, their water sources are most likely contaminated by many enteric pathogens. So kapag po itong si traveler nakainom siya ng tubig galing dun sa mga lugar na to, this individual will now develop diarrhea. And itong ETEC po yung strain that was predominantly isolated from those cases of diarrhea. So kaya po naging ganun yung tawag sa disease, which is again, traveler's diarrhea. The preferred habitat of ETEC in the human body is the proximal region of the small intestine. So also take note that people who are suffering from achlorhydria are the population who are most at risk to acquire infection from the ETEC strain. So, i-define muna po natin yun, no? What is achlorhydria? So, this is the absence of hydrochloric acid in the stomach. So, maliban po kasi sa digestion, hydrochloric acid in the stomach also helps us eliminate some pathogenic organisms, di ba po? Because some of those organisms can't survive at the extreme level of acidity. If you would remember, natutunan din po natin to sa parasitology classes nyo, di ba po? So some parasites that can usually be found in the oral cavity cannot be found in the stool, because in the event that the host swallows these parasites, by the time na nakarating po sila sa stomach, they would have already died or have disintegrated by exposure to stomach acids. Kaya po, hindi natin sila mahanap sa stool. So ngayon po, kung walang stomach acids in the case of people with achlorhydria, that's one less line of defense for the host to fight off both parasitic and bacterial pathogens. Next is the entero-invasive E. coli, or the EIEC. So this produces a dysentery-like or Shigella-like infection, which we'll get more into later once we talk about the Shigella species. The preferred habitat within the human host is at the intestinal epithelial cells. So kaya po tinawag na entero-invasive because it can penetrate or invade the intestinal cells. Next is the enteropathogenic E. coli or EPIC strain. So the enteropathogenic E. coli is the causative agent of infantile diarrhea. EPIC does not produce any toxins, but it can cause damage, which leads to the loss of microvilli, as illustrated here in this figure. Next is the enterohemorrhagic or EHEC. So enterohemorrhagic E. coli or EHEC. Specifically, EHEC serotype O157H7. So ano po ang meaning ng mga letters na to? So ito po yung serologic antigens na na-discuss natin kanina. So somatic O antigen 157 and the flagellar H antigen number 7. So the virulence factor of EHEC includes virotoxins 1 and 2 and also the Shiga-like toxin. So these toxins have the capability to destroy vascular endothelial cells, which is why these are the types of diseases that it can cause. So hemorrhagic colitis and also bloody diarrhea and the HUS or the hemolytic uremic syndrome. So HUS or the hemolytic uremic syndrome is a condition in which the small blood vessels of the kidney are damaged and inflamed. And then the last strains are enteroadherent and diffusely adherent E. coli or the EAEC and DAEC. So they have been documented to cause both urinary tract infections and also watery diarrhea. Next organism na po, Klebsiella pneumoniae subspecies pneumoniae. So the common name for this is the Friedlander's bacillus. This is the most commonly isolated species of Klebsiella. Now, Klebsiella pneumoniae subspecies pneumoniae is the causative agent of community-acquired pneumonia, which is characterized by expectorating a current jelly-like sputum. So currents, these are a type of small berries. So sinasabi po nila na yung sputum ng pasyente kahawig ng jam or jelly that is made out of those berries. So ang sosyal, di ba po? Sa Pilipinas, kung sa Pilipinas to pinangalanan, sasabihin lang nila mukhang ketchup. Ketchup-like sputum. Okay, so as I've mentioned earlier in the general characteristics, Enterobacteriaceae are non-encapsulated except for 2 genera, and that includes Klebsiella. So, ang Klebsiella po ay encapsulated. So, itong polysaccharide capsule po nila, this would serve as their virulence factor. So, in relation to that, dahil nga po encapsulated sila, it will be very obvious in their colony morphology. So, on McConkey agar, we can observe them to be pink lactose-fermenting colonies that are mucoid. Okay? Kahit po dito sa BAP, they always appear very shiny, almost kind of moist or wet-looking, and yun nga po, ang description natin dito is mucoid. Okay, so since encapsulated po ang Klebsiella, this will also show a positive reaction if we were to subject it to the Kellogg-Capsular swelling test. So, saan po natin unang na-discuss or na-encounter yung Newfield Kellogg-Capsular swelling test? During our discussion on Streptococcus pneumoniae, di ba po? So, kung nakalimutan nyo na yung principle and procedure of the Newfield Kellogg-Capsular swelling test, please do take time to review our discussion for Chapter 15. So, ito po yung mga biochemical test reactions ng Klebsiella pneumoniae. So, Klebsiella pneumoniae is indole negative, methyl red negative, which would automatically mean that it is Voges Proskauer positive, di ba po? As I've said from my earlier discussions, there is no clinically significant bacteria that can be positive for both MR and VP. And lastly, sila po ay citrate positive. Okay, and then if you would notice, magkabaliktad po sila ng reaction ng Escherichia coli on INVIC, di ba po? Ang E. coli, positive, positive, negative, negative. But for Klebsiella pneumoniae, the reactions are flipped. So it's negative, negative, positive, positive. One similarity that they have though is parehas po sila ng TSI reaction. Okay, so as you can see right here, parehas po sila na acid over acid. What? Acid over acid, tapos with a positive gas production, but negative for hydrogen sulfide production. And one reaction that would set them apart is that Klebsiella pneumoniae subspecies pneumoniae are also urease positive. Okay, so let's now talk about other Klebsiella species that have been associated with numerous infections. So first up is Klebsiella oxytica, which produces infections that are similar to those of Klebsiella pneumoniae. In addition, isolates have been linked to antimicrobial-associated hemorrhagic colitis. With regards to its biochemical test results, Klebsiella oxytica is identical to Klebsiella pneumoniae except for its production of indole. So kung makikita po natin dito yung INVIC reaction niya, for oxytica it's positive, negative, positive, positive. E di ba po for pneumoniae it's negative, negative, positive, positive. Next is Klebsiella pneumoniae subspecies Ozonae, which has been isolated from nasal secretions and cerebral abscesses. Okay, so this organism causes atrophic rhinitis, which is characterized by the inflammation of, or by the formation of thick, dry crusts in the nasal cavity, which has resulted from the progressive wasting away or decrease in size, which is termed as the atrophy of the mucous lining. So in general po, Klebsiella pneumoniae subspecies Ozonae has been linked to tissue destructive diseases restricted to the nose area. So upon exploration of the nasal cavity, this is what the infected areas would look like. Okay, so another thing that we have to keep in mind regarding Klebsiella pneumoniae subspecies Ozonae is that they are highly associated with the presence of extended spectrum beta-lactamases or plasmid-mediated ESBLs that contribute to the large numbers of antimicrobial resistant hospital-acquired infections. Ang mga ESBLs po, or microorganisms that produce enzymes known as extended spectrum beta-lactamases, are resistant to most beta-lactam antibiotics, including penicillins and cephalosporins. Next is Klebsiella pneumoniae subspecies Rhinoscleromatis, which was formerly referred to as a separate species called Klebsiella rhinoscleromatis, and this one has been isolated from patients with rhinoscleroma, which is an infection of the nasal cavity that manifests as intense swelling and malformation of the entire face and neck. Okay po, so this photo on the lower right shows us a case of a 63-year-old man that presented with a 10-year history of a mass growing in the central region of his face involving his nose and upper lip. So, hindi po nagpa-consult agad yung pasyente, no? Inabot ng 10 years para mag-progress or maging ganito kalala yung kondisyon ng pasyente. So, upon histological examination, it revealed that there were foamy macrophages, and the culture of tissue fragments showed the presence of Klebsiella. So again, that was for Klebsiella pneumoniae, subspecies Rhinoscleromatis. Next is the Enterobacter species. There are two common isolates for this genus. So Enterobacter aerogenes and Enterobacter cloaceae. So Enterobacter cloaceae and aerogenes are the two most common isolates from this group, and these two species have been isolated from wound infections, urine samples, blood, and even CSF. So they may resemble Klebsiella on MAC, and why is that? Bakit po kaya magkahawig yung colonies ng Enterobacter compared to Klebsiella? Kasi po, di ba, parehas silang encapsulated. So I didn't include a picture of them anymore to make space for this table on the right, but you could expect na pink, mucoid, and lactose fermenting din po yung colonies ng Enterobacter species. Even their INVIC and TSI reactions are the same. Now, if we would want to differentiate Enterobacter aerogenes from Cloaceae, then that is where this table will come in handy. So although they are both ornithine decarboxylase positive, lysine decarboxylase positive po aerogenes, but Cloaceae is negative. On the flip side, arginine dihydrolase is positive naman po for Cloaceae but negative for aerogenes. Alright, and then for other tests that may be used to differentiate these two species, you may check out table 19.6 from our reference textbook. So may extended version po ito sa reference textbook natin. Next is Serratia marcescens. This is an opportunistic nosocomial pathogen that has been associated with bacteremia in nurseries, cardiac surgery units, and burn victim units. So just almost anywhere in the hospital, really. So some strains of Serratia marcescens are late lactose fermenters. So recall time po tayo ulit. From what we have learned from chapter 9, which biochemical test should we perform to identify late lactose fermenters? The correct answer is ONPG, or the orthonitrophenyl beta-degalactoperoxide test, which is done to determine if a bacteria is a late lactose fermenter. Okay? So ayun po. Now let us look at its other characteristics. On McConkey agar, we may be able to observe its characteristic brick red colonies due to the production of the pigment protegiasin, specifically when incubated at 25 degrees Celsius. So, huwag pong malilito, no? Please do not confuse this with a positive lactose fermenting reaction. Kapag lactose fermenter po ang isang organism, it will produce pink colonies, and also there will be a pink precipitate or discoloration around those colonies. Pero itong pong colony morphology ng Serratia marcescens, walang precipitates sa paligid ng colonies, and also they're not really pink. They're described to be brick red, and this is because of the pigment protegiasin. So here are its INVIC and TSI reactions. So Serratia marcescens will also test positive for ONPG, gelatinase, DNase, and urease. Alright, so I have been following a few pages on social media that are dedicated to posting microbiology content, such as this one. So I just thought that I'd share it with you guys. Okay, so ayan po, this is a photo of Serratia marcescens that has been plated on BCYE, or the buffer charcoal yeast extract. So pwede rin po pala nating i-isolate ang Serratia marcescens using the BCYE. So it looks really nice, and I just thought that it would make a good transition to another recall challenge. Okay, so recall po ulit tayo. BCYE, or the buffer charcoal yeast extract, was originally formulated with L-cysteine to isolate specifically which genus of small pleomorphic gram-negative bacilli? Okay, so para po kanino talaga ang BCYE based sa natutunan natin sa chapter 18? Yes, this is for the isolation of Legionella species. Next is the Proteus species, commonly isolated from urine, wound, and ear infections. So they can also infect the proximal kidney tubules and cause acute glomerulonephritis, or AGN. So they are not simply just urease positive, but they have also been noted as a rapid urease producer. So here's the thing. When urease breaks down urea, it produces an alkaline urine pH. So ang normal pH po kasi ng urine is slightly acidic to neutral. But during Proteus infections, it raises the pH to be alkaline, which actually encourages the formation of some renal crystals, which eventually accumulate as renal stones. Now, in your future analysis of urine and other body fluids, or AUBF classes, matututunan nyo po that there are different types of urinary crystals, as well as at which pH environment do they form. Okay? So, some crystals would form in an acidic environment. Yung iba naman po, they don't care. Acidic, neutral, or alkaline man, pwede pong mag-form yung mga urinary crystals na yun. While some urinary crystals form in an alkaline environment. And one of those reasons that contribute to the formation of these urinary crystals that eventually turn into renal stones is, ayun nga po, yung alkaline environment that the Proteus species has created. There are many other species, but only these two are considered as human pathogens. So Proteus mirabilis and Proteus vulgaris. Ayan po. So, huwag kakalimutan yung mga characteristic colony descriptions nila. They produce clear, non-lactose fermenting colonies with a swarming motility on MAC, as we can see here in this picture. And even on BAP po, pwede pa rin natin ma-observe yung swarming motility nila. So, kapag po sinabi na swarming motility or nag-produce ng swarming phenomenon, ang isang isolate, dapat po, una nyo nang maisip is Proteus species. With regards to their odor, they have been described to produce a burnt chocolate or a burnt gunpowder odor. Okay? So, minsan po, kahit makalimutan nyo or makonfuse kayo sa mga biochemical test results nila, ng kahit alin sa mga bacteria na na-discuss natin, there will always be these couple of things that will still help us to recognize them. So, wag nyo pong kakalimutan yung mga may highlight na yan. Okay? Kasi, kahit po makalimutan nyo yung mga biochemical test results nila, if mababanggit po yung ibang mga characteristics na to, this could give you a good clue as to what organism is being asked. Alright? Here are the reactions for both Proteus mirabilis and vulgaris. So on INVIC, the V right here means that their reactions could be variable. Okay, so minsan magne-negative, minsan magpo-positive. Ang sigurado lang po tayong magkaiba sila dito sa INVIC reaction nila is their indole production. So mirabilis is negative, vulgaris is positive for indole production. Another test that we could do to differentiate them is the ornithine decarboxylase test. So mirabilis would test positive, while vulgaris is negative. Next is Morganella morganii, which may be very easily confused with Proteus vulgaris because almost all of their biochemical test results are the same. So ano po ang point of differentiation nila? Morganella morganii does not produce swarming colonies. And also, it is negative for hydrogen sulfide production. So, kabaligtaran po ng Proteus species, right? The genus Providentia is composed of 5 species, but only 3 of these are associated with human infections. So, we have Providentia alcalifaciens, Stewardii, and Red Gary. So, Providentia redgeri is a documented pathogen of the urinary tract. And then Providentia stewardii has been implicated in outbreaks in burn victim units and has also been isolated from urine cultures. So infections caused by Stewardii and Rickettsia, especially in immunocompromised patients, are particularly difficult to treat because of their resistance to antimicrobials. And listed here are their biochemical test results. Two notable species under the genus Citrobacter include Citrobacter furundii and Citrobacter kozeri. So Citrobacter furundii is commonly recovered from diarrheal stool cultures, and because up to 8% of Citrobacter furundii species can produce H2S, and then some strains of about 50% of them would fail to ferment lactose, the colony morphology of Citrobacter furundii on primary selective media can be mistaken for Salmonella when isolated from stool cultures. And then for Citrobacter kozeri, here they cause totally different clinical manifestations such as neonatal meningitis and brain abscesses that can be seen in CT scans such as these ones. Okay, so since yung mga Citrobacter po, they are late lactose fermenters, pwede pong magbago yung itsura ng colonies nila depending on how long you have kept it incubated. So within 24 hours, they would appear as your typical clear non-lactose fermenting colonies. But if we were to extend the incubation period for up to 48 hours, makikita natin na nagbago na siya. Okay? So Citrobacter colonies can now be seen as light pink lactose fermenting colonies after 48 hours of incubation. And this is attributed to the fact that, yun nga po, late lactose fermenter sila. Alright? So, dito po pala sa genus na to, kahit hindi tayo mag-ONPG test, i-extend nyo lang yung incubation period nila, malalaman na po natin na sila ay late lactose fermenters. Here are other biochemical test results of Citrobacter furundii versus kozeri. So on INVIC, isa lang po ang pinagkaiba nila, and that is the indole production. So Citrobacter furundii is indole negative, while Citrobacter kozeri is indole positive. Plasmonas was formerly under the family Vibrionaceae because of its phenotypic characteristics, but further studies showed that this is actually more closely related to Enterobacteriaceae. So one thing that makes it stand out from the rest of Enterobacteriaceae is that Plasmonas shigelloides is the only oxidase-positive member. Also, one of its similarities with Vibrio is that it is also sensitive to the O129 vibriostatic agent. So Plasmonas shigelloides is a straight gram-negative bacilli that is non-encapsulated, non-spore-forming, and it is motile. So they may occur singly or in pairs or in short chains of filamentous forms. So they may have monotrichous flagella up to, or they may also have up to 5 lophotrichous flagella. Okay? So recall po ulit tayo, recall time. Ano po yung isang genus ng aerobic gram-positive bacilli that has also been described to be branching or filamentous? So if you remember from chapter 16, diba, was it chapter 16, we talked about this one genus that was also filamentous. The correct answer is Nocardia species. Okay, so wag po kakalimutan. Nocardia species has also been described to be branching or filamentous upon microscopic examination, as we can see here in this pop-up screenshot. So, pwede natin siyang i-gram stain or pwede rin naman pong modified acid fast. And ma-observe natin in both stains yung filamentous forms ng mga, or filamentous arrangement ng bacterial cells nila. Alright, so balik tayo sa Plasmonas shigelloides. So the antigens of this could cross-agglutinate with Shigella sonnei, Shigella dysenteriae, and Shigella boydii, which is most probably why it was named Plasmonas shigelloides, because it has some Shigella-like characteristics. Cases of gastroenteritis due to Plasmodium shigelloides are usually self-limiting, and this is acquired most often after the consumption of undercooked seafood or intake of untreated water. So there are three major clinical types. So one is simple watery diarrhea. Two is subacute or a chronic disease that can persist from 14 days for as long as 3 months. And then the third one is the most serious one. It can be invasive and it may resemble colitis. So ano nga po ba ang ibig sabihin ng colitis? So colitis is characterized by the inflammation of the inner lining of the colon or a segment of the large intestine. Extraintestinal infections happen commonly to those who are exposed because of their occupation, and immunocompromised individuals, for immunocompromised individuals, systemic infections such as bacteremia and meningitis may occur. Plasmodium shigelloides will not grow on TCBS, while eosin methylene blue agar and McConkey agar may inhibit the growth of some strains. And then any other culture media is fine, but inositol, brilliant green bile agar is the most recommended one for the isolation of Plasmodium shigelloides, and their colonies will appear shiny, opaque, with a slightly raised center and a white to pink pigmentation. Its biochemical test results will help us in differentiating it from Aeromonas, Vibrio, and other members of Enterobacteriaceae. Okay, so while all the other members of Enterobacteriaceae are oxidase negative, Plasmodium shigelloides is oxidase positive. Next po, Aeromonas is resistant to the O129 vibriostatic agent, but Plasmodium shigelloides is susceptible. Now, both Vibrio and Aeromonas cannot ferment inositol, but Plasmodium shigelloides is an inositol fermenter, which is why its preferred culture media is the inositol bile green agar. And finally, as shown here on the photo to the right, Plasmodium shigelloides will thrive in a nutrient broth with 0% salt, but it will not grow on the same broth that has an added 6% sodium chloride. Okay, so do take note though that this is the same is true for Aeromonas. So Plasmodium, to be honest, its classification seemed very confusing for quite some time. So serologically, it could cross-agglutinate with Shigella species. And then biochemically, parehas naman po silang oxidase positive ng Plasmodium and Aeromonas. Pero hindi sila parehas na susceptible to the vibriostatic agent. But then, further studies showed that it's actually more closely related to Enterobacteriaceae, which is like, boy, pick a side. Let us now discuss the three primary intestinal pathogens, also referred to as overt or true pathogens. They are not part of the normal flora of the human gastrointestinal tract. In fact, they're not part of a normal flora anywhere else in the human body. And being able to isolate them from patient samples is considered as a very significant clinical finding. So the three members of Enterobacteriaceae that have been classified as true pathogens are Salmonella, Shigella, and Yersinia. Members of the genus Salmonella produce significant infections in humans and even in certain animals. So many Salmonella serotypes are typically found in cold-blooded animals as well as rodents and birds, which serve as their natural host. So according to Mahon, there was this time in America where a lot of people were documented to have acquired salmonellosis from their pet frogs. So, ayun po, Salmonella species in general are gram-negative, facultatively anaerobic bacilli that morphologically resemble a lot of the other enteric bacteria. Infections may be acquired either by ingesting contaminated and improperly cooked food or by direct contact with human carriers. So factors that are responsible for the virulence of Salmonella include Fimbriae, used in the adherence in initiating intestinal infection. Another would be enterotoxin production. So enterotoxin produced by some Salmonella strains that cause gastroenteritis has been implicated as a significant virulence factor. And lastly, Salmonella species also have the capability to move across the intestinal mucosa. Okay, so Salmonella species have antigens that are similar to those of other enterobacteria. Meron din po silang somatic O antigens and flagellar H antigens, and these serve as the primary antigenic structures used in the serologic grouping of Salmonella species. Pero po para sa Salmonella, their K or capsular antigen is otherwise known as the V or VI antigen. So the serologic identification of the VI antigen is important in identifying Salmonella serotype typhi. Okay, so currently, based on DNA homology and sequencing, the genus Salmonella comprises of only 2 species. So first is Salmonella enterica, and under Salmonella enterica are 6 other subspecies. Okay, so ayan po, anito yung Salmonella subspecies: enterica, Salome, Arizonae, Diarizonae, Hutinae, and Indica. Tapos po, within the Salmonella subspecies enterica, there are about 3 other serotypes. So serotype typhi, cholerae suis, and paratyphi. The next species in this genus is Salmonella bongori, which is a rarely isolated species that has been named after the town of Bongori, Africa, and this species was initially isolated from a lizard, and that was usually found among cold-blooded animals. 2 categories of infection. First is gastroenteritis, commonly termed as food poisoning. So listed here are its modes of transmission and clinical manifestations. Next naman po is enteric fever or the typhoid fever, which is caused specifically by Salmonella enterica, subspecies enterica, serotype typhi, or simply this is referred to as Salmonella typhi. So this is a febrile disease that can be acquired through any of the following ways listed here: ingestion of food prepared by infected individuals, also contaminated food and water, and direct transmission, or otherwise known as fomites. And then clinical manifestations include anorexia, myalgia, severe frontal headache, and the appearance of what has been termed as rose spots, which may be seen anywhere on the patient's body, and this is usually an indication of untreated typhoid fever. Okay, so as shown here on the right, this is how rose spots would look like. Salmonella enterica are hydrogen sulfide or H2S producers, but on McConkey agar, since wala naman pong H2S indicator ang MAC, we would only see them as clear, non-lactose fermenting colonies. Okay, pero kung sa SSA po natin sila i-isolate, the SSA or the Salmonella Shigella agar, which again is something we'll talk about at the last segment of this discussion, dito po natin makikita yung characteristic fish eye colonies nila, described as clear colonies with black centers, and this is because of their ability to produce hydrogen sulfide. Okay, and of course, as usual, listed here are its biochemical test results. Next is Shigella, and this genus is actually very closely related to Escherichia. Kaya po, diba, if you would remember our discussion earlier on the different strains of E. coli, yung EIEC, this has been described to produce a Shigella-like infection, and it is able to invade the intestinal cells. While yung EHEC naman po, on the other hand, this produces a Shigella-like toxin. So there are a few similarities between Escherichia coli and Shigella species. Now, on McConkey agar, their colonies look like any other clear non-lactose fermenting colonies, but on SSA, they also kind of look the same as Salmonella except that they don't have black centers kasi hindi naman po H2S producer ang Shigella species. Now, as we can see right here on this table, there are 4 different species, and they are classified into 4 different serogroups. So the basis of their serogrouping would be their somatic O antigen found on their cell walls. So Shigella dysenteriae is group A, tapos yung Shigella flexneri po is group B, Boydii is C, and Shigella sonnae is group D. So just try your best to not get them mixed up or try to make a mnemonic for this one. So nung time po namin, yung tinuro sa amin nung seminar prof namin was don't F boys. Okay? So this is a family-friendly channel. Bahala na po kayo kung anong ibig sabihin ng F, di ba? Basta don't F boys. So it stands for the proper order of the Shigella species. So dysentery is A, flexneri is B, Boydii is C, and sonnae is D. Let us first look at the general biochemical characteristics that is applicable to all the 4 species. Tapos tingnan natin kung alin po yung mga exemptions. So all Shigella species are mannitol fermenters except for Shigella dysenteriae. Next, all Shigella are negative for gas production except Shigella flexneri. So ang Shigella flexneri is positive for gas production. And then all Shigella species are negative for ornithine decarboxylase except for Shigella sonnae. So ang Shigella sonnae po is positive for ornithine decarboxylase. And lastly, all of them are negative for lysine decarboxylase. All of them. Walang exceptions para sa isang to. Now, on the second column, other specific characteristics per species have been enumerated. So first up is Shigella dysenteriae. So this is the most virulent out of all of them. And this is mainly responsible for causing bacillary dysentery. So ito po yung nagpo-produce ng Shiga toxin. And here is a list of its biochemical test results. And then in the past, Shigella flexneri has been noted as one of the causes of gay bowel syndrome along with Giardia intestinalis. I just got sidetracked. May nag-attend po ba dito ng qualifying exam review that was hosted by Ma'am Liz Segundo? Di ba po yung isang question doon na sinama ko sa parasitology review tungkol dito, hindi ko po sinasadyang nasabi yung sagot. Kasi tinanong ko kay Ma'am Liz kung yung Giardia intestinalis ba or Lamblia yung ginagamit nung time nila. So, ayun po. Wala. Naalala ko lang because of the gay bowel syndrome. So, anyway, if you attended that specific review, you know. If you know, you know. Meanwhile, ang Shigella sonnae naman po, it causes a self-limiting form of watery diarrhea. So this is also a late lactose fermenter and would definitely test positive for ONPG. Alright, let's now talk about bacillary dysentery caused specifically by Shigella dysentery type 1. So here are its modes of transmission, and its clinical manifestations include the non-specific ones such as fever, chills, abdominal cramps, painful bowel movement, and this may also cause rectal prolapse in children due to excessive straining of the rectum. Okay? So, just in case nakalimutan nyo na po, una tayong na-introduce sa condition known as rectal prolapse sa parasitology classes natin. So, Trichuris trichuria has also been documented to cause rectal prolapse among children, which happens when a portion of the rectum protrudes outside of the anus, turning itself inside out, as we can see here in this picture. Okay? Ayan po. And then, the more serious clinical manifestations would include acute inflammatory colitis. There's also bloody diarrhea, and this is also composed of not just blood, but also WBCs and mucus. So one of the complications of bacillary dysentery is the hemolytic uremic syndrome. Next is Yersinia pestis. So this is commonly known as the plague bacillus, and this is the causative agent of the bubonic plague, or the Black Death, that has caused the death of approximately 75 million to 200 million people across Europe, Asia, and many other regions during the 14th century. So Yersinia pestis has been classified as a type A bioterrorism agent by the Centers for Disease Control and Prevention. So recall time. Recall muna po ulit tayo. There are two other bacterial pathogens na na-discuss na po natin in the past. One was an aerobic spore-forming gram-positive bacilli, and the other was a gram-negative fastidious organism. So parehas po sila na listed as a Category A bioterrorism agent. Recall time, ano po ulit yung dalawang yon? Okay, so what were the other two pathogens that we've previously discussed has been classified as a Category A bioterrorism agent? The correct answer is Bacillus anthracis and Francisella tularensis. So, yung aerobic spore-forming gram-positive bacilli po is B. anthracis, tapos yung gram-negative fastidious organism naman po is Francisella tularensis. Ngayon po, itong member ng Enterobacteriaceae na to, which is Yersinia pestis, lahat silang tatlo is classified as a Category A bioterrorism agent. Okay, so nabanggit ko na rin po sa general characteristics kanina, di ba po? All Enterobacteria are motile except for Klebsiella, Shigella, and Yersinia. So again, Yersinia species is non-motile. And then the virulence factors of Yersinia would include coagulase and fibrinolysin. So una po nating natutunan yung mechanism of action and function nitong dalawang virulence factors na to when we learned about the many other virulence factors of Staphylococcus aureus. So, now, medyo naiiba po yung microscopic morphology niya from all the other Enterobacteria, because although, yes, this is still considered as a straight gram-negative bacilli with rounded ends, it has an odd staining pattern that has been described as a bipolar staining pattern, or a safety pin appearance, as we can see here in this photo to the right. Here is a list of its appearances on different culture media. Tapos ito naman po yung mga biochemical test results nila. There are actually two forms of the plague caused by Yersinia pestis. The first one is more popular. So this is the bubonic plague, or Black Death. And unlike all the other members of Enterobacteriaceae, ang mode of transmission po nito is not via ingestion. Rather, this is acquired from being bitten by an infected flea belonging to the species Synapsilla chopis. So this photo would show us the clinical manifestations. And if you would direct your attention to the bottom right image, kaya po siya tinawag na Black Death is because it causes necrosis of the tissues, turning it black once the infection has reached a septicemic stage. Okay, next po is the pulmonary plague. Ang pulmonary plague naman po, this only occurs secondary to the bubonic plague. At kahit wala pong flea vector, people could acquire this through close contact with other infected individuals. So again, that was for the pulmonary plague. Next is Yersinia enterocolitica. This is the most commonly isolated species of Neisseria, and it causes infections typical of an enterobacteria such as waterborne gastroenteritis or enterocolitis. Do take note, though, that this pathogen has the ability to cause an infection whose clinical manifestations can mimic those of someone who is suffering from appendicitis, hence the term appendicitis-like syndrome. Okay? So, ayan po, lagi natandaan kapag nabanggit na it may cause an appendicitis-like syndrome, dapat po, una nyo may isip is Yersinia enterocolitica. So now that we've established the fact that Yersinia in general are non-motile, another exemption is that Yersinia enterocolitica is different. Okay, so at 22 degrees Celsius, it can give a motile result in SIM. And then one selective media that can be used to isolate Yersinia is the cephsulidin ergocin novobiacin agar, or the CIN. So Yersinia enterocolitica will produce transparent colonies with dark red centers, and this has been described as bullseye colonies, as we can see here in these photos. So ito po yung itsura ng isang bullseye, and this is what the colonies of Yersinia enterocolitica would look like. Okay, so they do look pretty similar. On IMVIC, its indole production would be variable, but we are sure, though, that this is MR positive, VP negative, and citrate negative. Tapos, ito naman po yung mga TSI reactions niya. On this table, we can see the different TSI and LIA reactions that each members of Enterobacteriaceae can produce. So the problem here is that there are some organisms that fall into two or more different interpretations. So tulad po nito, yung Proteus mirabilis and Proteus vulgaris po. Ayan. So both of them has been noted to produce an A over A positive for H2S reaction sa TSI. But if we were to look over here, pwede rin po silang mag-produce ng K over A positive for gas and positive for H2S. So, di ba po, these are the same organisms, but they could produce different TSI reactions. Tapos, tuloy naman po tayo. Tignan natin yung Escherichia coli. So, E. coli over here could produce 3 different results on TSI. Okay? So, pwedeng K over A positive gas, or kaya K over A lang, or pwedeng A over A and positive for gas. Pero po, it can produce 2 different results sa LIA. So, pwede po siya na K over K, tapos dito rin po, pwede siya na K over A. So, sometimes it could be very confusing. So, kailangan po, makilala talaga natin yung bawat organism, not just based on their biochemical test results, but also based on their own unique characteristics. So, kung ano yung mga diseases that they cause, what do they look like on different culture media, and other stuff like that. Speaking of different culture media, let us now quickly go through the different culture media in the laboratory diagnosis of Enterobacteriaceae. First up is the McConkey sorbitol agar, or the MAC-SOR, or the SMAC for short. So this was formulated by removing the lactose and replacing it with sorbitol into the original MAC formula for us to be able to differentiate EHEC, or the E. coli 0157H7, compared to all the other E. coli strains. Kasi po, ang 0157H7, this cannot utilize sorbitol. Pero po, yung ibang E. coli strains, they can utilize sorbitol. So, positive isolation of 0157H7 would show us clear, non-sorbitol fermenting colonies. Okay, so if we were able to isolate just a regular kind of E. coli, then they would be able to observe pink sorbitol fermenting colonies. Okay, so kapag ganito po yung itsura, this is what all the other E. coli strains would look like. Pero kapag ganito po yung itsura, negative for sorbitol fermentation, then most likely po, O157H7 po yung isolate. The eosin methylene blue agar, or in some textbooks is referred to as the Levine's medium. This is used to differentiate lactose fermenting colonies such as E. coli that can produce their iconic greenish metallic sheen. So if the bacteria is lactose fermenting but it is not E. coli, then their colonies would appear purple, just like this one. So this is an example of Enterobacter aerogenes on EMB. Kapag naman po non-lactose fermenting, yung isolate natin, the colonies would appear colorless, just like these ones right here. And then other coliforms that belong outside of the family Enterobacteriaceae will grow pink-colored colonies as pointed right here. Next is the hectone enteric agar, composed of basalts and bromthymol blue dye. So these two are supposed to inhibit the growth of other gram-negative bacilli for better isolation of Salmonella and Shigella. So lactose fermenting enterobacteria would have yellow colonies. Tapos po, non-lactose fermenters would have blue-green colonies. Tapos depende na lang po if they produce hydrogen sulfide, then we would be able to see that they have black centers. Meanwhile, non-enteric pathogens will produce orange or pinkish orange colonies, just like these ones right here. So again, these are for non-enteric pathogens. Next is the Salmonella Shigella agar, specifically formulated to differentiate Salmonella and Shigella species from other enteric bacteria. So here are its components, and here is what Salmonella and Shigella would look like. So ito na po yung nabanggit natin kanina na fish eye colonies. So right here, this is what we would expect of Shigella, and ito naman po yung fish eye colonies ng Salmonella, because again, they are hydrogen sulfide producers. Last but not the least is the xylose lysine desoxycholate agar. And ang pinakakailangan lang po natin tandaan dito is that this is used for the isolation of Salmonella and Shigella pa rin, but from heavily contaminated samples such as stool. Here are the references that were used in the making of this discussion. That ends our lecture on Chapter 9. So for all the other miscellaneous organisms that were not mentioned in this presentation, I highly suggest that you read about them on your Mayhon. Thank you so much for listening.